So today we're going to be talking about the cell walls of eukaryotic cells. And they're actually of prokaryotic cells. They're actually very unique structures and molecules that are found in the cell walls of prokaryotic cells. And many of them are actually recognized by our own immune system. So our bodies know when we're being attacked by bacteria. Now, antimicrobial substances tend to target some of these structures. So that's going to be our focus. Now, there are two different types of cell walls, gram-positive and gram-negative. So we're going to have to distinguish between the two of them. Now, the most important chemical compound in the cell wall composition of these cells is called peptidoglycan. It is a macromolecule found only in bacteria that contributes to the rigidity of the cell wall. Now, it's only found in the eubacteria, those that we would consider humans to come in contact with more frequently. They don't necessarily live in extreme environments. So what we're going to do is you've got a table in your note packet, and we're going to go through and actually fill out the table, fill in the chart. So you can see on the left-hand side, there's the gram-positive bacteria, and on the right-hand side, there's the gram-negative. So what we want to do is distinguish between the two. Now, when we use the gram staining process, we can see both purple and pink bacteria if gram-positive and gram-negative are both on one slide. Gram-positive turn purple. I always remember that because they both start with a P. Purple positive, right? The pink is negative, and the reason I remember that is because pink has an N in it. Purple doesn't. So, I don't know, that's just how I remember it. Anywho, so when we look at these groups, the representative genera include Bacillus, Staphylococcus, and Streptococcus. So we had the opportunity so far in our lab to take a look at those. And so we know that many of those were purple when we looked at them underneath the microscope. Escherichia, Neisseria, and Pseudomonas are examples of genera that would stain that reddish pink color and actually be considered gram-negative. So what is the big difference between the two? Well, in the gram-positive, there is a really thick layer of peptidoglycan, really thick layer. So up here on this diagram, there's the cell membrane, and then there's the peptidoglycan, super thick, and that's what's in blue there. That is actually what we consider the cell wall of gram-positive cells, okay? Now, look at where the blue is here in the gram-negative. There's a really thin layer, and it happens to be sandwiched in the periplasm or space in between an inner and an outer membrane. Very, very different. So gram-positive bacteria have teichoic acids, gram-negative don't. But you look at the next three items, and gram-positive don't have any of them, but gram-negative do. That periplasm up here, that outer membrane around the outside, as well as this orange layer that's represented here. And that's the lipopolysaccharides, or what we typically refer to as LPS. It's considered an endotoxin and very dangerous to humans. Okay, porins are absent because there's no outer membrane, so they don't need special channel proteins to allow things to pass through, where they are present in the gram-negative. When we look at the sensitivity to penicillin, the most basic antibiotic out there, gram-positive are generally more susceptible, where gram-negative are not. Okay, we're going to talk about why that is here in a little bit. And in terms of sensitivity to lysozyme, lysozyme is an enzyme that's found in your saliva and tears. It's kind of one of your basic chemical defenses that your body produces naturally. And gram-positive bacteria are susceptible, whereas the gram-negative aren't. Okay, so what happens is the lysozyme or the penicillin both attack that peptidoglycan, break it down. Now when that happens to the gram-positive, the cell actually becomes a protoplast. And so one good example of that is when you're sleeping and you wake up in the morning and you get the crusties or the eye gooberies in your eyes. That's because overnight, while you were sleeping, your eyes create tears, right? And then that tear has lysozyme in it. And so it's basically destroying any of the bacteria that might be on the surface of your eye while you're sleeping. Gram-negative doesn't necessarily affect the peptidoglycan because it's sandwiched in there. It's protected between the inner and outer layers. All right, so let's look more specifically at the gram-positive. So the gram-positive cell wall has a big-time thick layer of peptidoglycan. And so that's represented here by these alternating turquoise and purple subunits called NAM and NAG, which are N-acetylglucosamine and N-acetylmuramic acid. Really long names. That's why scientists typically refer to them as NAM and NAG. Anyways, they alternate, and then there's I can't even think of the word I want to use. Cross connections between each chain, and that's what forms the different sheets of peptidoglycan. Now, there can be up to 30 layers of sheets in the gram-positive bacteria. That's a lot of peptidoglycan. But remember, beneath that, they only have the single cell membrane. So because all this peptidoglycan is on the outside, that's why it's susceptible to antibiotics and lysozyme. So you get infected with a gram-positive bacteria, no problem. Easy to address with antibiotics. Now, looking at the gram-negative bacteria, it's a little bit different. So remember, if I work my way from the inside out, there is my inner cell membrane, there's the periplasm, or a fluid-filled space, and then there's a couple of sheets of those alternating NAM and NAG forming the peptidoglycan. Then there's the outer membrane that has the lipopolysaccharides associated in it. Okay, so now we're looking at 3 layers: inner membrane, peptidoglycan, outer membrane. Kind of a big deal, big difference. So when we look at the gram-negative, let's write down that there's a thin layer of peptidoglycan, only 1 to 2 layers. It has an inner and outer cell membrane that contains the peptidoglycan plus the LPS. That gives us our 3 layers. And it is not susceptible to antibiotics and lysozyme. Now, the periplasm, so that you can write this down as a definition so you don't forget, is a fluid-filled region between the inner and the outer membrane where that little bit of peptidoglycan is located. The lipopolysaccharides, or LPS, are toxic and are considered an endotoxin to our system. So when the body is attacking the bacteria, as the bacteria break down, the cell wall breaks up, that LPS is released and it gets into our bloodstream. Now what that does is it actually causes a person to go into shock and eventually into septicemic shock. So it's something that you can die from, unfortunately. Now, I talked a little bit about penicillin and lysozyme in the chart. Well, exactly how does that work? Penicillin interferes with peptidoglycan synthesis. So new bacterial cells can't be made. So when you think about binary fission, a prokaryotic cell is reproducing. It's going to create 2 new daughter cells. But before the cells can literally split apart, that cell wall has to be made. Well, if there's no peptidoglycan being made at the ribosomes, the cell wall can't be made. Therefore, the cell never actually splits apart. So penicillin literally stops an infection by keeping the cells from reproducing. Lysozyme, on the other hand, literally destroys peptidoglycan. Doesn't matter if it's a reproducing cell or not. Okay, it breaks the bonds between the NAM and the NAG. Sorry, the dog and cat are playing. Between the NAM and NAG in that peptidoglycan. And so it destroys any bacterial cell. Doesn't matter if it's reproducing or not. So what happens if bacteria don't have a cell wall or peptidoglycan? Well, then penicillin and lysozyme aren't going to work. So for our purposes, we want to know, how can we figure out if somebody's got a gram-negative infection? Because if that's the case, then they're susceptible to developing shock. So how do we test for that? Well, if we get a sample of fluid and we're able to culture the bacteria, we can perform what's called the gram stain test on it. It's a real basic procedure. It's a differential staining process that was developed by Hans Christian Gram over a century ago. It allows us to distinguish between 2 different groups of bacteria, obviously gram-positive and gram-negative. Now, let's look at the first step. First, we've got our slide with the bacteria on it. We add a primary stain called crystal violet. It turns all of the cells on the slide purple. Doesn't matter if it's gram-positive or gram-negative. Everything turns purple, okay? As a matter of fact, your fingers will turn purple if you get it on you. Second step, we add iodine. Iodine acts like a mordant, almost like a sealant, so the cells remain purple. They don't change color because of the iodine, but the iodine, the mordant, actually combines with the crystal violet and peptidoglycan to create an insoluble compound in the gram-negative bacteria. So to create a difference in the cells, we add alcohol, which kind of acts like nail polish remover. It takes the purple out of the gram-negative cells so that they become colorless. Now, because the gram-positive have that diiodine complex present that's insoluble, the purple doesn't come out. So at this point on our slide, if we've got both gram-positive and gram-negative present, we're going to have purple cells and clear cells. Well, we want to be able to see the clear cells. So the final step is for us to add a secondary stain. And that secondary stain then colors those gram-negative cells a reddish, pinkish color. Now, think about it. The secondary stain is pretty important. If I just added purple again, I still wouldn't be able to tell the difference, okay? But because I'm adding a lighter color, it doesn't change the purple in gram-positive, but it does make it easier to find the gram-negative. So my question at the bottom of the screen is, why is this important in medical microbiology? Why do we need to know if someone has a gram-positive or a gram-negative infection? Well, if somebody's got a gram-positive infection, we know we can administer antibiotics. If they have gram-negative, we have to be careful, because if those bacteria are broken down and the LPS is released, then the person can get septicemic shock.